The next speaker is Professor Miguel Holgado. He's professor here from this school, from ETSII Industriales, and he's also the member of Centro Laser, or Laser Center, if you want in English. And his field of interest is biosensors and optical sensors. So we are going to hear something about this topic as well. And Miguel, when you feel ready.
Okay, so good morning. First of all, thank you very much. I would like to thank the organization for giving me the opportunity to make this presentation, especially Teresa, in this annual meeting, in this 7th annual meeting. And okay, I fear that my presentation will be quite different to the other ones. So okay, I will try to explain in which we are working on since 2007. Okay, just firstly, just starting by saying that we are producing or developing photonic structures for sensing, not only for biosensing, also for gas sensing and liquid sensing. Our idea is just to produce this kind of a structure. This is the first slide here, that can enhance the response of some kind of sensors. After that, we read out these kind of structures for producing some readout signals in order to convert these physical phenomena in readable information. So, okay, because I think that probably in this case it's important to make an introduction of which is a biosensor. And after that, I will just explain which is our strategy for label-free biosensing. And in the several slides, I will just explain you how are working our biosensors, how can we produce this kind of biosensors, and something very important, how can we read out information of these kind of structures. Finally, I will just give you some examples about some immunoassays and biosensing examples in which we are working on right now, and in other kind of information. So, which is a biosensor? Okay, well, a biosensor is like a sensor in which there are some kind of bioreceptors. Bioreceptors normally is an antibody that we mobilize in our sensing surface. These bioreceptors normally capture selectively some kind of biomolecules. These kind of biomolecules, or target biomolecules, we call them antigens. So typically proteins, hormones, viruses, or even bacteria, or even ADN. So once that these captures or these bioreceptors captures the target biomolecules, in our case, the sensing surface change the mass, change the refractive index. In another kinds of transducers, this kind of recognition produce a mechanical signal, electrical signal, and so on. In our case, this change in the mass, a slightly change in the mass, produce a change in the transducing signal. I don't know if I have, yes, that one. Yes, as you can see here, this is an optical response in which we are representing here, for instance, the reflectance versus the angle of incidence, depending on the readout system that we use. So these patterns give us the information about what is happening here. And then what we are working now is just in produce a kind of optical transducers to produce that this signal changes a lot when this recognition effect happens. There are right now, two kind of strategies for in vitro diagnostic. The standard one is based on label strategy, in which here are the steps. The first steps always is just to immobilize a bioreceptor in a sensing surface. After that, the antigen or the target biomolecule will be captured by this antibody. And in the case of label-free biosensing, in which there is any kind of chemical amplification, so this is really a challenge just to detect this kind of interaction, we produce some photonic structures to detect what is this interaction. In the standard test, when you are going to the clinician or where you make some blood analysis, what's happened is that there is another antibody, which is called secondary antibody, and this antibody normally have an enzyme, and there is a kind of chemical amplification producing, for instance, fluorescence, luminescence, or change the color. But our work is just to make sensors for detecting this direct interaction. So based on this principle, the kind of, we can just produce that an antigen have a kind of size, the molecules have a size, which is in the range of nanometers to several nanometers, or even in the case of 1 micron or more in the case of bacteria. But normally we use our biosensor just for this dimension, just for 1 nanometer to 100 nanometers. So you can see that it's quite difficult to recognize selectively and detect small molecules in the order of 10 nanometers, which is the typical size of a protein. And which is our vision, or how has been our strategy for level 3 biosensing? Okay, the first thing is that to see small things, we need small sensors. So we thought that producing this kind of nanosensor, which is a nanopillar, when the light interacts with this nanopillar, the changes caused by the effective refractive index on the sensing surface drastically change the response of this kind of nanosensor, because here we have a kind of interference or resonance or something like that. The problem is that to read out a nanosensor is quite difficult. So our idea was just to produce a network of nanosensors. Therefore, we can read out the contribution of all of the nanosensors, producing something that we call B-cell, biophotonic sensing cells. So then this kind of biophotonic sensing cell, we can produce these biophotonic sensing cells with a size of 10 hundredths of micron, 60 hundredths of micron, or 10 microns, so it's just depending on our design. And then that we have a biophotonic sensing cells, we can produce chips. These chips have a number of this kind of B cells, or biophotonic sensing cells, and then we have a biochips where are capable of detecting this kind of biomolecules. And after that, we produce biochip kits, and after in this biochip kit, for instance, we are just attach many of our biochips, and therefore in this kind of kit we have available 100 diagnostics just in a single kit. So this is something that is not currently in the market, and because of this idea we set up a new company that is called Biooptical Detection. So why B cells? So the good thing is that if we compare the standard response of a single interferometer, here in this curve I'm representing the blue signal without any kind of biomolecules inside of the sensors, and the red one is when there is a small bifilms, there is a films biomolecules just on the sensing surface. And here we can see the difference when this is happening. And if we produce our biosensors based on nanopillars, you can see that this pattern, this profile, drastically changes, enhancing the sensitivity. And you can see here that these two patterns, if we compare the difference of the distance in the of the spectrometry profile, because we are representing here the wavelength as a function of the reflectance as a function of the wavelength, you can see here that this displacement of the dips or peaks are much higher than in this case. So now then we have some kind of parameters, of dimensional parameters for our designs. And then depending on the diameter of the pillars, the lattice parameter, I mean the distance or the different pitch among pillars and the height of the pillars, we can produce different kind of biosensors. But how is really working this? Okay, in our week we are covering all the value chains from the basic research, just considering which is the interaction in this kind of our sensors. So in fact, the light when interact with this kind of B cells, the light is just coupling independently in each of these nanopillars, just is covering, is just traveling through the pillars, And there is a kind of there is an electric field because the light is not only inside of the pillars, it's also outside. We call this a evanescent field. So the electric field just traveling onto the sensing surface produce a change in the refractive index, and we are detecting this. Depending on the interaction, I mean that depending, for instance, of the as you can see here, of the pitch among the pillars, we can enhance the electric field onto the sensing surface, and therefore we can improve the sensitivity. As you can see here, the typical, one of our typical simulations. But this is not only a theoretical one. You can see here a real and actual beam comparing with our simulations. And you can see that it is quite similar, the simulation that we are carrying out with the experimental results. And I will show you later on how is the final response in a practical way. So, therefore, just considering the structure of our B cells, of our biophotonic sensing cell, changes these parameters, we can see here how the signal are changing. Just by reducing the pitch among the pillars, we can see that these signals are changing and producing a much better sensitivity, just measured by the distance between the dips and the peaks with and without biomolecules, as you can see here. So then we thought about, okay, why pillars? Why not other kind of structures? This is something in which we are working right now, because as far as we know, if we made this kind of structures, there are some gaps, there are some areas, geometrical areas, where the electric field is much higher, and therefore, because the biomolecules are just onto the sensing surface, we can enhance the sensitivity. So we are working now in this kind of structures that we have characterized, and we have simulated that this one will have much better response than the single pillar. And the first experiments corroborate this one. But another thing is, why not resonant nanopillars? So we are producing this kind of pillars, and each of the nanosensor is a Bragg cavity. It's a resonant nanopillar. This will enhance the Q factor of the response, as you can see here. Of course, we can enhance the electric field just in the areas when we have to recognize the biomolecules, and produce this kind, theoretically, we produce this kind of response. But this is not only theoretically, but also you can see here the first experimental results of this kind of B cell based on resonant nanopillars. This has been also patented, and then something important, and there will be the next step of my presentation, is, okay, we are producing wonderful transducers, but another important thing is how we can read out these kind of transducers. And then you can see here some sensitivity maps in which, okay, we can measure the ellipsometry, the spectrometry, or the reflectometry. Okay, before to explain this, okay, if I remember well, okay, we are producing our own structures. We are using a photoresist, and we are also fabricate our sensors scalable at wafer level. As you can see here, we are using, for instance, mass projection lithography or laser interferometry lithography, or we are collaborating with other groups with e-beam lithography or other kind of technology. But very briefly, then we have a mask. We design our mask. Then we make a standard step of photolithography, make many sensors and many dies here. We cut the dies and package these biosensors or these chips just onto a chip. And also we have produced one of our readout or point of care platforms for readout the chips. Okay, based on our designs, we produce this kind of B cells. There will be a bit quicker. So something important, as I told you before, is not only the response of the transducer itself, but also the way in which we can read out the B cells. And then we have many standard technologies based on reflectometry at profile level and ellipsometry at profile level. This is very important because from the basic research, we can obtain optical information in a very small areas, in an area of less than 1 micron. Therefore, we can also, right now, because this is very new, analyze even a single nanopillar. Afterwards, we have a high-resolution spectrometry based on the standard gratings or interferometers. And you can see here, this is this equipment, which is just for basic research, has been under our design. This one probably the one which is capable right now to evaluate a single nanopillar has been designed by ourselves, and now we are working with this. And therefore, this is a very, very important information for designing also our response. And here, this is very, you can, it probably is not well, you cannot see very well the image, but this is the first images of the first responses that we have obtained with this kind of equipment, in which we analyze the spectrometry profile just in a single picture. This is where I think that we can apply with your embeddable system. So these kind of profiles give us a lot of information. Give us submicrometric ellipsometry, reflectometry, depending on the state of polarization, and producing also this kind of pattern for biosensing based on ellipsometry. But this is very well for the lab, but this is probably quite difficult for a commercial equipment or something like that. So then we produce this kind of point-of-care device, and I will show you a video if I have time, in which you will see all this process. With which point of care, just we are reading many visuals, just implemented in this kind of kits, and then you can see here some response. And now, very recently, we are working in a new methodology in collaboration with CEI, in collaboration with the group of Teresa, in which we are defining another kind of sensing strategy for readout the systems based on the autocorrelation function and comparing different kind of interferometers. Based on this idea, we are right now working with the CEI, and then we have produced, we have made the first test performing very well. So Mavi is the responsible for that, and very well, really good work. So just give you some example for diagnostics. We are starting with our vessels obtaining a really promising results with a limit of detection in the range of 3 nanograms per milliliter, just with a BioSci model, BSA, anti-BSA. BSA is the bovine serum albumin. This is the typical, the most frequent protein that we have all the human beings and also the animals. Also, we produce our vessel just in transparent substrates, just not only for evaluate the response of the biosensing just in reflection, but also in transmission. This gives us the possibility to make another kind of strategies. And now we are working in many applications. This is very important, and this is why I make the question about the site of the drug, because right now we are working with these companies to detect some kind of diseases in the dry eye. This person we have the dry eye disease. So we are evaluating six biomarkers just in a single drop of 1 milliliter, because something with dry eye disease is very difficult to obtain drops with a lot of volume. So now our technology permits to do this, and you can see here a kind of experiments certifying that we are working in a good track. Also, we are working in infection diseases with these companies for dengue virus, which is it's not very important in Spain, but is a very important disease around the world. Also for rotavirus, which is rotavirus cause the double of deaths in the world than the SIDA, than the AIDS. So it's also very challenging information. And just for the conclusion, say that our transducers based on resonant nanopillars were selected for this European project, in which we are working in detecting biological risks for aquaculture. So it's a very big project in which we are involved. Here are the funding agencies in which we are, most of them. And I think that, our conclusion is that we think that we have developed a simple biosensing technology for high performance and low cost in comparison with the standard one based on level technology. I would like to put a video. It's very brief video.
It's very brief, but I think,
But probably it's better than the questions, I don't know. No, I can answer all the questions. Okay. It's just to show the, okay, this was our okay, it's a bit old probably video, but it's important to, I think it gives you an idea about what we are working. So here are our kits. Probably I can go. Then just very briefly an explanation about how we can produce our chips. It's just a photolithography step in this case. Then we produce the wafer. In the wafer we have many dies, and therefore we have many chips. Then we cut and obtain the biochips. And then just only to give you that is, in our case we can work just with drops of the analyte. So that means that we can just deliver some drop of the analyte that we have to detect in this kind of wells. And in this well we have available many sensings or many diagnostics. Then we clean the chips, and then we put this in our point of care device. There are many positions for reading out the systems, and then we obtain the response with some. You can see here that this is a whole development, and okay, and here we have the final response and so on. So probably if you have any question, you will have to do it just after the presentation because there is no time. But in any case, if you have anyone.
Thank you very much for your really interesting presentation. I'm going to allow just one really short question because we are running out of time and we are eager to have a coffee. So please, one short question. Okay, I have one question, really short. I mean, we have seen all those pillars that you have to produce. Is it important to have them uniformly distributed, or I mean, do they have to be grown in the really same way? I mean, is there the sensitivity of your sensors? Does it depend on?
Yes, depends.
How do they grow? Or
yes, okay. Yes, it's a really good question because there are right now many kind of nano wires and so on, which is random ones. The problem with random ones is that each of them have a different response. So therefore, you cannot standardize that. When you have a wonderful networks, you know that the response will be always the same. It's not depending on the fabrication.
Yeah.
And therefore, that's the reason why it's important the order.
Okay, thank you very much.